Hello, everyone. Thank you for joining us today webinar. My name is Chin. I'm Technical Application Specialist, Dolomite. So today webinar is one of the webinar series we call Microfluidics 101 with Dolomite. This delivering every last Thursday of each month along 2021. So today topic we will learn about the liposome and lipid nanoparticles generation. Today, agenda will be including a presentation, and after that, we're moving to the Q&A section. So if you have any question, please feel free to write down the question in question box or chat box. So I will try to directly answer to all of you in the Q&A section. The content for presentation today will be including the introductions and the microfluidic technology for liposome or lipid nanoparticles generation. And then we will learn about the controlling the synthesis of liposome or lipid nanoparticles, and system scale-up and flexibility, and finally the conclusion. So the first introduction. So the liposome or lipid nanoparticles are spherical nanoparticle composed lipid with one or more bilayers. So depending on type of lipids, we can generate different types of nanoparticles like liposome or lipid nanoparticle or lipids. So the liposomes were discovered in 1960s, and the hollow nanoparticles are phospholipid vesicles consisting of at least one lipid bilayer. And this bilayer is usually composed of the amphiphilic phospholipids that have a hydrophilic phosphate head and a hydrophobic tail, which consists of the two fatty acid chains. So these structures feature had facilitates liposome applications, including the use at artificial cell membranes, carry for drug delivery systems, encapsulating agents for food ingredients, and analytical tools. Liposomes also are widely employed at drug delivery systems. So using the liposome or lipid nanoparticles, there's a lot of the advantages as so in here. So the first is flexible for hydrophilics and hydrophobic drug encapsulation. And liposomes are also non-toxic, flexible, biocompatible, and completely biodegradable, with non-immunogenic for systemic and non-systemic administrations. And liposome also increase efficiency of therapeutic index of encapsulation, and reduce the toxicity of the encapsulate agent. Also, liposome can reduce the exposure of the sensitive tissue to toxic APIs as well, and with the side avoidance effect. And finally, is the flexibility to couple with the site-specific ligand to achieve targeting. So the reasons why nowadays using the liposome or lipid nanoparticles is the hot topic in pharmaceutical and other applications. So we know that with the onset of the COVID in 2020, so the world had realized that infectious diseases are very much first world problems, and nanoparticle space, especially around the liposome and lipid nanoparticle, has exploded. So the lifespan of the disease is directly linked to the introduction of the vaccine. Also, with the COVID-19 has by far broken records for the fastest vaccine development, and most, if not all, vaccine require packaging before being administered with the lipid nanoparticles or liposome are by far most popular delivery vehicle. So actually, in May 2021, the Google search for lipid nanoparticles or liposomes will it grew from 1 to 2,000 searches per month in 2020 to 15,000 searches per month globally. So if you have a look at the breakdown of nanoparticle type, is the FDA approved or in clinical trials. So we can see the liposome and lipid nanoparticles have most highest numbers of nano formulation in used in nanomedicine. So there's unmet needs in drug delivery and life science markets. So the next, we will move to how to use microfluidics technologies for liposome and lipid nanoparticle generation. So with lipid nanoparticle or liposomes, is the formers using a process of the nucleation and growth. So the lipids will be dissolved in solvents, for example, like ethanol or other alcohols, and hydrodynamics flow focused in the chip by anti-solvents like waters, phase, or the buffers solution. So the lipids nanoparticles begin to assemble at the results of the mixing, and it's believed to form at the interface of the two streams and flushing out by the continuous stream. So the particle size of the nanoparticles is controlled by the adjusting, changing the flow rate ratio and total flow rate. So for the encapsulation, the API can be brought in good continuous phase for hydrophilic APIs or interface for hydrophobics, or also it can be encapsulated afterwards through the active loading methods as traditional method. So using the microfluidic technology, it brings many advantages for liposome or lipid nanoparticle formulation with high encapsulation efficiency, with high, very excellent PDI, with rich reproducibility at low sample volume, and it's easy to scale up. As using microfluidic, we can apply very small amount of energies in the process. So if you have a look at this image showing the microscope images of two streams of the liposome to form the liposome by using the lipid solution and the aqueous phase to generate the focused flow focusing stream between the two phase, and using two different the geometry of the chip design. Here's a single channel system for liposome or lipid nanoparticle synthesis. So basically, the system will be including the three pumps to driving the fluid or reagent into the microfluidic chip, which located under the microscope integrated with the high-speed cameras. So the systems were designed very flexible. So using the different chips, you can achieve the different application. And for liposome and lipid nanoparticles generation, so using the different chip type geometry, so we can generate a different range of the nanoparticle size and also the different production rate or throughput of the nanoparticle generation. So as you can see in this image, we can use a different chip geometry to obtain the different kind of flow rate, different production rate, and particle size range. So we can use the three flow focusing chip, or we can use the 5-in-2 chip, or we can use a micromixer chip with the micromixer channels to improving the micromixing in the process. So this one showing the system setup for a single channel system. So the lipid and ethanol will be pumped using the one pump and pumping to the interface in the channels of the microfluidic chip. So in here, for example, we use a 5-input microfluidic chip design. So the lipid and ethanol will pump in interface in here. As you can see more detail in here. And the others aqueous phase or buffer solution will be pumping in the outer phase, outer channels line in here. And you can see in here connecting to this one. And it will be generate the flow focus stream at the chip junction to be generated the nanoparticles, lipids or liposome nanoparticles. So for particle side controlling, we adjusting of the flow rate ratio between the interface and outer phase. So with the flow rate ratio increase, you can see, so we can obtain the different particle size. Increase flow rate ratio, we can achieve the smallest particle size. Here is just showing some results with our system using the different chip. So for this one, we use a 5-input chip with 150 deep channels with the pore. So the condition is shown in here: 10 milligram per mil for polypon and DDAB is 2.5 milligram per mil as the surfactant in ethanol, and aqueous phase will be PBS solution with a pH 7.4. So the results as showing in this graph, the total flow rate will control at 100 microliter per minute, and we can achieve the particle size range from the 100 to 250 nanometer by changing flow rate ratio from 510 to 80 value. So as you can see, when we increase the flow rate ratio, we can achieve the small size of the liposome or lipid nanoparticles. And we also achieve the very small value of the PDI values. So the value is lower than 0.2. And here is another result that we obtain when we using another chip geometry. So we use the micromixer chip in this with the same condition as the previous experiment. So as the results can show in this table, you can see when we do the total flow rate as a higher value at 500 microliter per minute, and the result showing is a very good low PDI values, and the particle size range from 60 to 120 nanometers by changing flow rate ratio. So using micromixer chip, we can obtain the smallest range of size, particle size, and also we can obtain the higher production rate as higher as the throughput of process. The other system that I would like to introduce to generate the liposome and lipid nanoparticles is we automate nanoparticle systems is for protocol development. So in here is basically how the system look like. It including the 2 quad pump, a searing pump, 2 searing pump with 4 independent channels that can working independently, and we have a pressure input store for the reagent containers, and we have the ultimate sample barrel with the sample loop to introduce the small amount of the reagent for the applications. And the collectors will be including the ultimate collector with the wire rack, so it for automatically the collection sample collection. So the system is designed together with two chips, one chip for the nano formulation process, and the second chip will be for the dilution process. The software controls the reagent injection loop, which allow the series injection of sample, thus allow you to do the protocol processing with the optimization condition, and automate the collection sample, automate the setup and run of the sequence of sample. So in this module, this system, the only manual step requires the loading of the lipid and cargoes into the loop in here. So we have to use the syringe and hand to inject the sample into the sample loop. So the others function like pump operation, loop switching, collection, washing, all will handle automatically by controlling using the software and protocol. And the system allows you to introduce a very small amount of sample from 200 microliter to big amount of sample like 5 milliliter per sample. And also with the system, after the protocol development or optimization process, it allows you to switch to continuous mode, production mode for batch production at up to 1 liter per hour for scale-up production. So the ultimate nanoparticle system is able to effectively compress of the stream of API against the center of filament of the lipid solution. So when using the hydrodynamic flow focusing method to make the liposome or lipid nanoparticles, so it's the interface between the central lipid stream and the surrounding the aqueous fluid, where the lipid nanoparticle or lipid molecules begin to cell assembles and eventually form the liposome. So using the different chip geometry, we can have achieved the different the targets, like using the T junction is for flow impingement and the flow focusing to fluid stream, or using the micromixer chip using the turbulent for turbulent flow mixing with increased mixing ability, and therefore you can achieve with the smaller range of the particles size and with the higher throughput of production rate. So using 3D flow focusing chip or 5-input chip, so you can get a better encapsulation of the API result. Here is showing the diagram for how the system works. Basically, we have 3 pumps. So one pump have one pump for driving the one nano precursor one, with basically it's a buffer solution in here for outer channel to outer channels, and the second pump we're pumping for the second precursor like lipids and ethanol, and we introduce to interface inner channels in here. This first chip. And the product will be go to from first chip to the second chip for dilution. So for dilution, the third pump will pumping for using for drying fluid for the dilution process with the T junction chip. And finally, the product will go to the collected sample and controlled by collection valve to decide go to waste or for collection. So the next section, the next slide, we will learn about how to control the synthesis of liposome and lipid nanoparticles using the automated nanoparticle system. So as we know, there's many variables that can affect to lipid nanoparticle or liposome synthesis. So the first variables is the formulation variables. For example, like type of lipids or concentration of lipid or ratio of lipids. So from that a variable, we can generate a different type of the nanoparticles, different size of the nanoparticles, and with a different application. And the second variable is the chip type, chip geometry. So it affects to the mixing the region. So it will be affect to also nano formulation process. And the third variable is the flow rate ratio. Is the flow rate ratio between the organic and aqueous phase. So with the changing flow rate ratio, we can we can obtain the different particle size distribution. And the fourth variable is the total flow rate. It's the combined total flow of the organic and aqueous phase. So with the total flow rate, we will affect to the particle size range or the production rate. And finally, the variable is the volume. So with nanoparticles, the precursor loading volume, or it collected sample volume, or it had a tail cut, or it the dilution. So all of the variables will be responding on the regions like lipid nanoparticle size, PDI values, and encapsulation efficiencies. And also, what is the important about the lipid nanoparticle and liposome that customers want to achieve? So the first important thing is the monodispersity with low PDI is good, low CV is good. And the second is the size control. So with the wiring of the targetable particle size is good for the customers. And the third one is consistency from optimization process to manufacture. So with rapid optimization particles synthesis during the R&D stage, so with minimal development required to pilot and manufacturing scale production is preferable. And number 4 is encapsulation efficiency. As a high number of the drug loading or the encapsulation efficiency with the minimal waste is targetable for lipid nanoparticle production. And finally, the control of payload release with tunable and reproducible payload release over time. Here showing some results to generate, to formulate the lipid and liposome and lipid nanoparticle using ultimate nanoparticle system. So in here is the condition for our experiment. So we use the same organic phase, same material with using in single channel system. So with the phospholipid and the DABs in ethanol, and aqueous phase will be PBS solution. With dilution is also using the PBS solution. The flow rate ratio will be variable in this experiment, and total flow rate will decide at 3 ml per min. So the results showing the system very easy to control over lipid nanoparticle size by just changing flow rate ratio. As you can see, when we increase the flow rate ratio in here, we can achieve the lipid nanoparticle size from 20 to 140 nanometer. And it achieved reproducible results, as you can see in this here, the graph showing the lipid nanoparticle produced at the fixing flow rate ratio 3:1 at the total flow rate 3 milliliter per minute, with the repeat 3 times with the average lipid nanoparticle size of the 134 nanometers, with the PDIs below to 0.1 value. So it targets low PDI values. And this slide showing the results to provide the maintenance of consistencies when increase the throughput using the automated nanoparticle system. So in this one, we use the condition is the same with the flow rate ratio will be 2:1:1 into figures here, figure 2, and 1.5:1:1 in figure 3. And with total flow rate will be changed from 1 to 10 or 1 to 15. So the results showing when we increase the throughput by increase the total flow rate. So there's no compromise in reproducibility. So as the increased total flow rate is symbolized less effect on the particle size distribution. So the particle size almost same when you keep the constant of the flow rate ratio and just increase of the total flow rate for increase of production rate. So the PDI showing the value is also very consistent. So average PDI of 0.19 with 0.01 standard deviation. And when you increase the cycle time, therefore also the increase libraries generation. So is very useful for using the protocol process. So in this slide, we're showing the development and production of the lipid nanoparticle liposome with a single system solution in here. So we show you the protocol mode to develop the nanoparticles formulation libraries with when you turn to protocol mode to continuous mode for bulk production, so optimized formulation using the same system. So it's showing the results. So it's very consistency, no compromise in lipid nanoparticle size, and PDI shipping from protocol to continuous production. So in this figures, you can see the lipid nanoparticle produced continuously total flow rate 3 ml per minute with the changing of the flow rate ratio. So lipid nanoparticle will be range from 19 nanometer to 57 nanometer. So when you can see there's no change between when you do the protocol mode and continuous mode, so it's very consistent when turning from the optimization condition to production scale. So the next part, we move about the system scale up and flexibility. For scale up system, again, as the previous the webinar, I already introduced. We have a Telos High throughput system, which is very flexible in use for application. So it's not only for using to introduce to generate the other particles like PLGA or a microparticle microsphere, but also it can be used for high throughput liposome or lipid nanoparticle production. So as we know, the production rate will be increased or scale up of system based on two way by using the Telos system. So it increases the number of junction chips working in parallel. So in chip we have in one chip we have 7 microfluidic channels that work in parallel. So that allows you to do at a higher flow rate, and it also increases the flow rate through the individual junction. So when we can integrate up to 10 chips working together in parallel in one system, so it allows you to increase the higher production rate. So this showing how the multiple Telos module can be stuck together to build up a system for larger scale production. So this image is showing the clamp module Telos. So it's using for one microfluidic Telos chip. So we have 7 channels inside. So we by this way without any tool. So it's very easy to quick to set up and use. And it can integrate up to 10 Telos modules. So it identical condition up to 7 microfluidic chip droplet junctions working in parallel together, which allows you very high production rate in the scale up process. And the chip working and controlling the individuals, so it's a fast-acting valves which allows one chip to be replaced without affecting the other. So if one chip getting problem like blocking or working unstable, so you can switch the valves, adjust module or just the clamp module, so replace this by new chip without any affecting to the other process, to the other chip and modules. Here showing some the results using the Telos High throughput system for lipid nanoparticle or liposome synthesis. So in here, this graph, you can use we use with the average Telos micromixer chip, as you can see in images with the micro mixer channels in here. So the total flow rate was studied at 5 microliter per minute. So the results showing that the particle size was successfully controlled in the region of from the 100th to 200 or 180 nanometer by just changing the flow rate ratio. So when you changing the flow rate ratio, you can as normal you can achieve the smaller particle size. So the PDI value also small. It's less than 0.18 in this case, and total flow rate can be up to 100 milliliter per minute by using the multiple module with multiple chips working together in a system. So it means you can generate a ton per month for liquid solution in production scale process. So this slide, I would like to introduce about our production scale development with GMP scale-up. So currently we stay at the production the protocol development system with the automated nanoparticle system in here. So we are developing the early system. It's a fully walk away automatically the system. So that allows you to do the more the automatically the processes protocol with the system, and you can see in here. So it's very it's a more candidate for API or lipid nanoparticle generation. And we also are doing the initial scale up with GMP certification with the ANPS system in continuous mode, production mode. And in production scale, we call it Titan with GMPs. So now several users are working to qualify the automated nanoparticle system for GMP production at 10 liters per day scale. And we have a roadmap to create from 10 to 100 times scale potentially based on our proven Titan platform. And we want to work with the partner company to define the scale and UI as required. Here is showing short introduction about the Titan platform that we are developing for production scale up. So it's an optional production scale system with from 0.5 to 1 liter per minute production scale. And we have successfully used this system to generate a lipid nanoparticle liposome and other nanoparticle type as well. So basically, as you can see here, is including 2 sealing pump just with one can allow you to running at the maximum of 250 milliliter per minute with 1 channel. So with 2 channels, 2 pump, you can pumping at maximum of 500 milliliter per minute or 0.5 liter per minute. So that's is for using for the production scale process. So here is the integrated of the reactor that allows you to generate the nanoparticle formulation at the high total flow rate, high production rate. And system flexibility, as I mentioned, the system is not only used for one application. It can be used for others, many applications with the flexible in the system setup and cheap using cheap geometry. So in here is one example to show the system flexibility in the liposome generation, like we can do with the Zen liposome, not only nanoparticle, not only nano liposome. So in this case, we can we do 2 processes. So the microfluidic our system will be stay at the step 1 of the process. So the step 1 we will generate of the droplet of the water in oil, so as a lipid at the using as a surfactant. So we can generate of the water droplet with the surfactant lipid layers, one layers cover outside of the droplet and disperse in the waters by using the droplet tip in here. So after the first process development, the droplet generation, so the product will be moved to the step 2 as the showing in here. So it will gently pour on top of the preparation of the solution that contains 1 layers of the lipid here, and it will be the inverted emulsion droplet heavier than the oil that contains them and segment into the second water phase, and they pass through the interface where the second layer lipids is sitting. So the bilayer is complete and the final vesicle forms. So you can generate of the Zen liposome just including the 2 lipid bilayers with contain core, the water core inside in micro side. And the other particles can be generated with the same system as we can do with the alginate, with the core-cell PLGAs, as we already or PLGA microparticle, as we already studied in previous last webinars. We can make up others like solvent harsh, the polyacrylamide bead, or PLGA nanoparticle as well. It's used for many applications like drug delivery, drug encapsulation, and pharmaceutical food industry, or screening R&D. Okay, so I'm finished the presentation today. So thank you for the attention, and we will move to the Q&A section right now. So if you have any question, please write down your question in the question box, so I will directly answer to you. Thank you. Okay, I got several questions in here. So the first question is from Zen Bass. Okay, so are the lipid nanoparticles produced in suspension form, or do you dry them to become powder after process? Yes, the question, yes, So the lipid nanoparticle produced with this method in suspension form. Finally, we will obtain some nanoparticles with content of the solvent and ethanol and the buffer solution or aqueous phase. So after post formulation, we can dry to generate to achieve the dry liposome by using the traditional methods like like evaporation process, normal evaporation process at low temperatures. So as you can use for in lab or R&D with the lab equipment. Also, the next question is also, do you remove the organic solvent ethanol after process? If yes, how? Normally, in lab, in our trials, the experiments, so we do not do the drying and remove the solvent. However, so the removing the solvent and ethanol and water is an easy step, as you can use. As I mentioned, it's very popular and using in you can get a reference in literature. It's very worldwide. So normally the people use the drying process. That's simply the evaporization process. So for example, like you can do it in vacuum condition to reduce the temperatures, to reduce the vaporization temperatures. So finally, you can remove the ethanol and water, as it's very easy to evaporize. I hope this helps. The next question is from Pretty. So how the number of channels in microfluidic chip can affect the flow rate and particle size? Actually, the number of channels in microfluidic chip can actually with the single channels in here. I guess that you are interested on the multiple channels like Telos chip. So in one single channel, we do which one maximum flow rate is depending on the viscosity or the precursor and the reaction condition. So it will generate the kind of back pressure and limitation in the flow rate. So when you increase the channels, micro channel in parallel, so it can allow you to do the process in the parallel condition. So it will allow you to increase the production rate, flow rate, total flow rate of the process. And for the particle size, it will do not affect too much. Actually, it will affect on chip geometry and the flow rate ratio, but in total flow rate, as I show in the experiment results, so the total flow rate doesn't affect strongly into the particle size. So mostly we control the particle size by just controlling the flow rate ratio between the two solvents, two streams, and also by using the different chip geometry. I hope it helps. Another question: there are different type lipids like single chain and mixed mono, di, and triglycerides. Which type of lipid would you suggest for using a microfluidic system? Well, so it depends, and also it's not limitation in using a different type of lipid for using the microfluidic system. So it depending on your application. So you can choose what type of lipid you use. So with the microfluidic system, so it just allows you to do the process with in micro stream. So that can allow you to do the fast mixing process and generate of the nanoparticle. So it can do with any precursor active type lipid. So it's strongly just depending on your application. Okay, so I got another question from Kajars. What's the cost of this instrument if we buy in India? Actually, the cost of this instrument is depending on the different what type of instrument that you are interested in. For example, is a single channel system and ANPS system, automated nanoparticle system. So we have a distributor, our distributor in India. Just you, if you are interested, we can send your address or let the distributors we will contact you to know and we'll let you know exactly what the cost of the systems you are interested in. So basically, it will be around in UK. Actually, in UK team, I'm not sure about the India. It's strongly depending on the market, India market. But in UK, so the system will be range from 30,000 pounds to 60,000 pounds. It depends on what kind of the system required, like single channel or ANPS, or what how many chip type you want, or how many modules systems that you want to use for your application. Okay, another question from Balasur, sorry, I cannot say Balasubramanian. Okay, so how do you control the size and molecular weight of PLGA simultaneously? Oh, yeah, this one is the question from the topic that we run as the last webinar. So for today is liposome, but PLGA, the particle size. The control is similar controls the way to control the particle size similar with in liposome generation. So you can control the particle size by just adjusting flow rate ratio of the two stream, outer phase and continuous phase, the interface. So by just changing flow rate ratio, you can changing the PLGA's nanoparticle size. Also, I don't know, you mentioned about the PLGA micro size or nanoparticle size, but in nanoparticle size, the same way with the liposome and with the micro size. So the PLGA was synthesized by creating a droplet between the PLGA precursor and the DCM, which is dispersed in the water. So after solvent extraction or solvent evaporization, so it will leave the PLGA contain microsphere. So the controlling of the PLGA microparticles by just controlling the droplet and controlling of the concentration of the PLGAs. So by both way. So for example, if you get bigger droplets, it means you can contain in one droplet more PLGA amount, so it will generate the bigger PLGA particle size and weight and higher particle weight. So the controlling of the droplet size is also using the changing or adjusting a flow rate ratio between the continuous phase and droplet phase, and or you can changing the chip junction size of the chip, your chip, microfluidic chip. So bigger chip junction size can generate a bigger droplet, small chip junction size, small droplet. So two factors: one, droplet size, and second is the higher concentration of PLGA, but not too high because the PLGA has a high viscosity if you're running at a high concentration. So the limitation may be around 10 to 15% of the PLGA in DCM. I hope this helped. Okay, one more question from Pretty. Can we avoid using organic solvent? How would it affect with the size of the particles? What do you mean about the organic solvent? So we use it on the ethanol as the alcohol in the process in our process for generation of lipid nanoparticle. So after processing, we can easily to remove the ethanols and water to generate the dry liposome. So it's generally using in pharmaceutical industry. So with the size particle size, it's also if you use different solvents like another alcohol, so it also will small effect, a little bit effect on the particle size. So, but as I mentioned, the particle size for the process is mostly strongly affected by the changing flow rate ratio. So during the process, the changing the flow rate ratio, you can achieve the different particle size. Next question from Si Han Wang. How can you encapsulate small molecules nucleic-based ingredient at low temperature? The system have temperature control. With a single channel system, we have a measures to control temperature. We call it a temperature control unit. So it can control the process from temperature from 1 degree C to 100 degree C with 1 degree C accuracy. RAM rate from the 1 to 100 degrees C. So it's very easy to control the process using that unit with very easy to integrate with the single channel system. And for ANP system, the ultimate nanoparticle system, nowadays we are also using the temperature control units for control the temperature in your tubing and your microfluidic mixer chip using the hot plate adapter, temperature control adapter. So it's very useful when you want to control the temperatures at your process, and also you can increase, you can control the temperature of the precursor before going to syringe by using the temperature bed, temperature controls like water bed. So it's very helpful for your temperature control process. So now we are developing the temperature control for the sample loop that can allow you to control the temperature, your reagent with contained in the sample loop. However, it's not released at this moment. It's in process, so we will release it very soon. I hope this helps. Is this possible? Another question from Rakesh. So is it possible to integrate your system to other systems like that potential particle size measurement process or the or other system? Yeah, actually, it's the zeta potential or particle size measurement process system is the kind of post analysis system. So there's no problem to connect, to integrate our system with this system. So the output of our system just only the one tubing just contain the product stream. So you can dilute the sample by just using our one of the pump channels and another chip for dilution and connecting directly to the post analysis equipment like the particle size measurement, or otherwise you can do it by just a batch method, like just to select sample by automated collected sample and to do with the post analysis. Otherwise you can online or inline to connect with the other equipment as well. It's no problem. I hope this helps. Okay, so I got one question from Niam Chan. sorry, if I do not pronounce right your name, Yim Chen. Okay, so what's the highest concentration of lipids the chip can process? So basically, we do the highest concentration at 10 to 20% is weight percent, molecule weight percent of the lipid source, which is dropped in ethanol. So I think it worked perfectly. So if you have interesting on this, we will send you some our application note that we describe more detail about the concentration of presentation, concentration of the lipid source to generate the lipid nanoparticle. So I will send you through the email after the meeting for your reference. Another last question I can see here is from Pretty as well. Can we control the temperature in the microfluidic process? Is this manual or we can input in the system? Yes, we can control the temperature in the microfluidic process. As I mentioned, we have the TCU units, temperature control units, so we can control your temperatures at the chip junctions from 1 to 100 degrees C, but it automatically controlled by using our protocol or our software. So it's very easy to input in the system, as our system very flexible and modular. So that is one modular in the system, so we can add it easily in our system, and it's very easy to control the temperature during your process. Okay, this is the last question I can see in the question box. Did you have any other questions? Otherwise, if you do not have now any question now, you can send the questions later through the emails that we already attached in our chat box, hanoioffice@blacktrade.com. So we are happy to answer all of your questions through the email as well. And before today's closing today's webinars, I would like to thank you everyone to join today our webinars, and just let you know that we will run another webinars, and next month, last Thursday, the next month, with the topics how to control and synthesis of the double or multiple emulsions using microfluidic. Is also very interesting topic that you can use in complex application like encapsulation, drug encapsulation, or vaccine development, or another food development, another process as well. Yeah, so if you have interesting on the topic, please go to our website and do the registration. So I'm very happy if I can meet all of you again in the next the webinars next month. Thank you very much, and have a good week, have a good day for everyone. Thank you.